Hello everyone, this is K 2023 Life Sciences Section P Biochemistry, question number 38. Enzyme activity profiles as a function of time in absence or presence of different types of feedback mechanisms as shown in the figure below. So these are the four different enzyme activity profiles, 1, 2, 3, 4. Match the following feedback mechanism with the corresponding profiles in the figure. So there are four different mechanisms. So P is without any feedback mechanism, no feedback mechanism. Q is negative feedback mechanism with a short delay. R is negative feedback mechanism with long delay, and S is positive feedback mechanism. So these are the different options A, B, C, D. After matching the enzyme activity profiles with these mechanisms. So let's first discuss what are the feedback mechanism. Okay. Also to mention, this is the NPTEL online course reference in which the feedback mechanisms and different types are explained. So coming to feedback mechanism. So feedback mechanism can be of majorly two types. We have a positive feedback mechanism and we have negative feedback mechanism, which is pretty simple to understand. Let's take an example. So we have a node A. Okay, in a network we have node A, and this node A follows a path to reach node B. This is path followed is known as a vertex or a vertex. It reaches to B in this direction. Okay, and these types of graphs are known as directed graph. Okay, in the directed graphs we have vertices going to a specific direction. Okay, so a simple example of a feedback mechanism is when a vertex or a vertex from a B reaches to node A, and this mode of vertex can be either positive or negative. Okay, so let's first take positive feedback, in which suppose node A is activating node B and node B further activates node A in a positive manner. This example is of positive feedback mechanism. Okay, this is the example of a positive feedback mechanism. Whereas in negative feedback mechanism, if a node A activates node B and further this activated node B inhibits, so this is the hammerhead arrow which represents inhibition, and this inhibition is negative feedback mechanism. Okay, pretty simple. So positive feedback is that node A activates node B, and after activation of node B it further activates again node A, and the cycle continues. Whereas in negative feedback mechanism, when node A is active, it activates node B and there is a negative feedback loop. Node A and node B are forming a loops. That's why they are also termed as negative feedback loop. And B is inhibiting A, which will then again inhibit B, activation of B. Since A is inhibited, A can no longer activate B, and thus B will also get inhibited. So this is a classic example of a negative feedback loop. Okay, so coming back to question, we have 4 different mechanisms. So P is no feedback mechanism. Okay, Q is negative feedback mechanism with short delay, R is negative feedback mechanism with long delay, and S is positive feedback mechanism. So first I'll explain this different profile. So on y-axis we have enzyme activity, on x-axis we have time. So suppose in this example, if we see that, if we give a signal, so before giving any signal, the enzyme activity is basal. So this is the basal. Okay, approximately 0 enzyme activity at basal level. So if we for our convenience, if we say at basal level, no enzyme activity, no enzyme activity. And as you can see, the graph is pretty straight line. There is no increase in enzyme activity, and as we gave a signal, the enzyme activity shoots up, okay, towards a shoots up to maximum. So suppose I added a signal for the this is 1 2. Suppose the time is in hours, so I added signal for 1 hour. After 1 hour, I gave a signal for an hour, and the enzyme activity stays stable. Okay, and then the signal is removed. After 2 hours, the signal is removed, and as you can see, the enzyme activity is still persistently stable and it is not falling down, right? So the enzyme activity is saturated and there is no decrease in enzyme activity, right? So even if I remove the signal, there is no effect on the enzyme activity. This is very important, that if I remove the signal after 1 hour, right, after 1 hour of signal treatment, if I remove the signal, the enzyme activity is somewhat remains constant. So we can say that even if we remove the signal, there is activity which is still persisting, right? So suppose node A, in the given example, node A is activating node B, right? And node B activation leads to activate A again, right? So we can say that from this example that we have given a signal, okay, which was activating A, right? And this A activated B, and then B activated A. When I stopped giving this signal, right? So I started giving signal at 1 hour and I stopped giving signal at 2 hours, right? But still, so I stopped giving signal, but still we are seeing the activity, right? We are seeing the activity of B. Why? Because when we provided the signal for this amount of period of time, we had activated molecules of B. We had various activated molecules of B, which further activated A molecules, right? And the enzyme activity persisted, right? So this profile, enzyme activity profile, corresponds to positive feedback mechanism, right? Clearly from this clearly explains the positive feedback mechanism. Okay, so so in the options S1 we have 2 options, B and C, right? So we are confirmed that it could be either B or C. Whenever there is match the following MCQs, first, if you are 100% sure about one option, then first see whether those options are MCQs, and if there are only one, then that is the final answer. You no need to solve all for everything if you are confident enough. So next, so these so the enzymatic profiles 3 and 2 are pretty complex, so we'll get back to them after some time. So let's look at enzymatic profile 4. So similarly, if we assume these are the 2 time points, so in enzyme activity profile 4, what we see is I am giving a signal at 1 hour, okay, and the enzyme activity shoots up, it reaches maximum, and till the signal is there, right, till the signal is there, it is staying at maximum. And once I remove the signal, if I remove the signal after 2 hours, the enzyme activity is it's dropping down, okay. So this is like when we switch on the lights. So we have a on button, right? To switch on the lights, we have on button. So we click on the on button. So the analogy here, what I am telling you is, so we have to keep pressing on this on button to keep the lights on, right? Once we remove the pressure on this on button, the light will switch off. This is what we are seeing here, right? We are giving signal from 1 to 2 hours, and as soon as we remove the signal, the activity is again at the basal level, okay. So there is no any kind of feedback or any activation or inhibition seen here, right? So P is 4, right? Pretty simple. No feedback mechanism. So in option B also we have P4, right? No feedback mechanism. Now coming back to these two profiles, profile 3 and profile 2. So I'll explain you what is negative feedback with short delay and negative feedback with long delay. So what is negative feedback mechanism, which I had explained before? We have a molecule A which activates a molecule B, and this B further goes and inhibits A. So indirectly we are saying that the B, so indirectly we can say that molecule B is inhibiting itself. Why? Because it is inhibiting the activity of A, and this activity of A will now no longer activate B, okay. This is the signal which was activating A. So in case of short delay, suppose this is a cell and these two molecules are present inside the cytoplasm. So what happens in the short delay is the inhibition response from B to A is short or very quick. The inhibition response is quick without any delay. When we provide a signal to A without any delay, there is inhibition from B to A, right? So this type of negative feedback is known as short delay, okay. Pretty simple. So why this is short? Because these two molecules are already present inside the cytoplasm along with other molecules, right? And there is quick response from B to A, and the inhibition is short. Whereas in long delay, what happens? We have a molecule A which is present in the cytoplasm, and then we have a molecule B. For example, it is present in the nucleus. When we provide a signal to the molecule A signal, then A is activated, which further goes and activates B. Okay, and then B inside the nucleus is only activated through the process of transcription or translation. So these processes takes time. Okay, there is a delay in this processes. So transcription takes around 30 minutes approximately. Translation takes around 1 to 3 hours depending upon the protein. So A rapidly diffuses and activates B, and after activation of it activates B via process of transcription and translation, which takes time. Okay, and then the production of B also takes time. So this B is delayed. B is delayed in its inhibition to A. Okay, so the inhibition which is provided from B is delayed by various processes. This is a general example. So this type of negative feedback is long delay. Okay, the negative inhibition, feedback inhibition, the inhibition takes over a period of time. It is not as quick as in the short delay negative feedback. Okay, so coming back to our profiles, remaining two profiles. So we'll start with enzyme activity profile number 3. So what is happening here? Suppose so we provide the signal at 1 hour. Okay, and this is the 2 hour time points. So we provide the signal at 1 hour. The enzyme activity is shooting up. Okay, this is indicating that A is activating B, as I mentioned in earlier examples. After giving a signal, A is activated, which is activating B. And as you can see, there is a inhibition in the enzyme activity because of a negative feedback. Okay, because of a negative feedback to B. So the enzyme activity is reduced. Okay, and these are the oscillatory behavior of the enzyme activity, and after a certain point this oscillation gets saturated. Okay, it gets saturated, and as the signal is removed, the enzyme activity suddenly drops. Okay, so this type of behavior is seen in short delay. Why? The inhibition is very quick. That is why we are seeing a rapid decrease. Yeah, we are seeing a decrease and the oscillation, and it is not again going back to its highest enzyme activity. The enzyme activity is decreasing because the feedback inhibition is a short delay and it is a quick response, right? The short delay means it has a quick response, and hence it is getting saturated over a period of the signal strength, and then finally it is going down. So Q is the third enzyme activity profile. This is the profile in short delay. Okay, whereas what happens in long delay is we have a signal. Okay, at 1 hour we gave a signal to A, it is activating B, and hence we can see a huge activity rise in the enzyme activity rise, and there is an inhibition which further activates B, and there is inhibition, so it is again going down. Okay, and because of it is going down, Okay, and the delay and the feedback is delay in the inhibition, and because of this delay there is again sharp rise in the enzyme activity, okay, to the maximum point, and after the delay it is again inhibiting the A, and again it is going down, okay, and this is continuous, and this is the oscillating behavior without any decrease in the enzyme activity. It is going to the maximum, it is dropping back because of the delay, and this type of oscillatory behavior, this oscillatory behavior is seen in long delay, because there is a delayed feedback, there is a delayed negative feedback mechanism to A, which leads to the rise in the enzyme activity, okay, and after the delay it is again decreasing, okay. So this type of activity is shown by long delay. If we match the options, the mechanism to the enzyme activity, the options, the correct option will be B, where P is where the so this is without any feedback, no feedback, without any feedback. This is how the enzyme activity profile will look. It's like we are giving a signal, and once we stop giving the signal the enzyme activity is decreasing, like on and off, right, on and off switch, and thus there is no feedback. Whereas we see a positive feedback, wherein when we give a signal, on signal, even after removing of the signal there is still persistent enzyme activity, okay. This is because of a positive feedback. We have a positive feedback here, right, and here the oscillations are dampening. This dampening of oscillations are seen in short delay, okay. So third profile is Q, short delay, and finally we have oscillatory behavior seen in case of long delay negative feedback mechanism, okay. So the correct option is B. Thank you.